predicts the evolution of the anatomo-functional organization of the primate prefrontal cortex.
Good afternoon, I'm Celine Amiez, and today I'm very pleased to present you some data showing that the Sulcal organization predicts the evolution of the anatomo-functional organization of the primate prefrontal cortex. If you look at the prefrontal cortex across different species from cat to man, we can see that in fact it's one of the structure that displays the largest expansion, especially in human, where it covers about 30% of the cortical surface. And if we look at the organization of Sulci, although you may have an impression that the organization is arbitrary, in fact, it's absolutely not the case. If we look at here a map from Michael Petridis, we can see that all Sulci in which you can see a little red label, in fact, they are found variably in the human frontal cortex. For example, we can see here this sulcus represented on two examples, that is the central sulcus, this one that is the Sylvian fissure, this one that is the superior precentral sulcus, the inferior precentral sulcus, the superior frontal sulcus, and the inferior frontal sulcus. Although you can see some variability, in fact, they are there in our brain. And it's true, and sorry, and last example, the intermediate frontal sulcus there. So even if you go further anterior, it's the case. It's the same thing on the medial, in the medial view on the human brain, when you can find the cingulate sulcus in blue and the paracingulate sulcus in 70% of hemisphere in pink. And it's also true in macaque. Here you can see variability in the sulcus, but if you look at that, you always have this sulcus in the macaque brain. And here the central sulcus again comes back on the medial view. You can see it there. So the first question I ask is whether the organization of the Sulci can provide a tool to understand primate brain evolution. Can we go from this macaque organization to this complex human organization? So to assess this point, we analyzed the anatomical MRI data on 197 human brains from the Human Connectome Project database, 225 chimpanzee brain, thanks to a collaboration with Bill Hopkins, 88 baboon, thanks to a collaboration with Adrien Megardichian, and 80 rhesus macaques that we collected around in the lab in Lyon, and also some external one. All these brains, they went through a linear registration to their respective template. For human, it was the MNI template, for example. So like this, you have the anterior commissure, posterior commissure lines that is horizontal in our brains. So let's start by looking at the Sulcal organization in the dorsomedial frontal cortex. So this blue area there. The first thing that we have demonstrated is that this paracingulate sulcus that we find in human in about 70% of hemisphere, and that is linked to many different aspects of cognition and personality traits specific to human. In fact, we discovered that you can find it also in chimpanzee brain. But the only difference is that it's less present. It's about in 30% of hemisphere. And whereas in human, it's left lateralized, it's not in chimpanzee. Baboon and macaque, by contrast, never present a paracingulate sulcus. If we look at the vertical paracingulate, vertical sulci that are present in the medial wall, in human, they are always sulci. The same thing in chimp, always sulci. In baboon, we see this type of shape that are what we call spur. And in macaque, you can see changes in the cingulate sulcus here, and that we call dimple. Okay, so what we can see here right away is that baboon and macaque, they present precursor of what we see as sulci in human. But if we look at the occurrence of this vertical sulci, and if we start from the more posterior one, the paracentral sulcus, we can see that it's equally present in human and chimp, and less, but still really often there in baboon and macaque. And if we look at what they look like, they look at sulci in human and chimp, mostly spur in baboon and mostly dimple in macaque. The pre-paracentral sulcus here just in front is the same thing. Equally present in human and chimp, almost always there, a little less frequent in baboon and macaque, but very present. And the same organization, the same sulcus spur dimple distribution is seen across the different species. The sulcus most anterior here, that is the posterior vertical paracingulate sulcus. You can see that it's very present in human, less in chimp, and almost equally present in baboon and macaque, but really low frequency. And we can see again that it's a sulcus in human and chimp, half-half spur or dimple in baboon, and always a dimple in macaque. And finally, the anterior one here, anterior vertical paracingulate sulcus, you can see a decrease of its frequency of occurrence across the different species. And same thing with sulcus. It's a sulcus in human and chimp. It's mostly a spur in baboon and a dimple in macaque. And now, so this shown that macaque do possess all the precursor of the human vertical sulci in this region. If we look at now where they are located, and you can see that the paracentral sulcus here is tightly linked to this anatomical landmark that all primates do possess, that is the rostral limit of the pons. Here, if we see here the distribution of the anteroposterior y-axis value, where we find the packs compared to where we find the rostral limit of the pons. So you can see it's well aligned in all species. The pre-packs is aligned on the anterior commissure. The P and the A vertical paracingulate sulcus are respectively aligned on the caudal and rostral limit of the journey of the corpus callosum. So this shown that all the vertical sulci are located at anatomical landmarks that all primates possess. Now, if we look at the sulcal organization of the ventromedial prefrontal cortex, this region is characterized by the presence of three sulci. The superrostral sulcus, that is very preserved one, always there in all species. The inferior-rostral sulcus, that is present in about 30%, 40% of hemisphere in human and chimp, and almost never there in baboon and macaque. The accessory susorbitalis sulcus here, that is that is present equally in human and chimp, almost always there, and still there in baboon and macaque, but less frequent. So this data show that all sulci in the VMPFC are sulci, even if macaque. No spur, no dimple, so it's a very preserved sulcus. And whereas the superior rostral sulcus is present equally in all primates, the two other ones are equally present in human and chimpanzee, but less present in all wild monkeys, but they are still sulci. And now if we look at this region that makes the junction between the dorsal and the ventromedial frontal cortex, we can see that it's in fact here that we can detect more expansion. If we look at what this region look like in human, this region always ends with the cingulate sulcus always ends by a fork composed by two sulci, the susorbitalis sulcus and the suprarostral sulcus here. When there is a paracingulate sulcus in human, this fork most often ends the paracingulate sulcus. Okay. And this fork in human is always pointing downwards, like this. Now, what's going on in the other species? So in human, we can see here it's a complete fork, always pointing downward. In chimpanzee, we see two cases, a complete fork pointing downwards, like most of the time, and a complete fork, but forward facing, like this. In baboon, we have four, and in macaque also, we have four patterns. We have these two patterns that we just show, like in baboon, a complete fork here, and forward-facing fork here. But we have also two other cases, an incomplete fork with only the susorbitalis sulcus present or no fork. And in macaque, we have the same pattern, but not in the same proportion. We can see here the gradient. The fork from human to macaque is, from macaque to human becomes more complete and points downward. And if we look now where this fork is locating on the dorsoventral axis, we can assess the intersection points here between the cingulate sulcus and this fork with the apex here of the journey of the corpus callosum. So the dorsoventral difference. And we can see that in fact in human, it's very close to this apex. Whereas in chimp, baboon and macaque, they are located further dorsally to it. So it means that the fork through evolution is pushed down in human. And this is linked to the expansion of area 9 and 10. So these data show that the macaques do possess all sulcal precursors than human in the medial frontal cortex, and that the most expanded region is this anterior one at the junction between the cingulate sulcus and the fork. And this the organization of sulcus so tells us about how the brain evolved, but it's interesting also to see that the sulcal pattern is linked to function. So if we look as an example, the relation to function between sulcus and function that we have in the mid-cingulate cortex, that is this region here, we have great evidence, similar evidence from human and macaque. In human, we have seen that when you do fMRI and you have subject to do simple task, motor task, movement of their mouth or their eyes, there is a face motor representation that is located in the rostral cingulate zone. And this is located here. So this is at the intersection with the posterior vertical paracingulate sulcus here in human, close to the caudal limit of the genu of the corpus callosum. And if there is no paracingulate sulcus, it's exactly there, but at the junction with the cingulate sulcus. So we have a face motor representation that is located here. And if we do a more cognitive task, that is the you are in a non-routine situation, you have to learn by trial and error which one of the different target is associated with the reward. They can be visual or just reward, and you look at how you analyze feedback to adjust your trial and error learning. And you look at this analysis of feedback during the exploratory search compared to when you repeat simply a choice that you know what the correct target is. This is also this same region that is involved. So we know that here we have a region involved in the analysis of visual juice feedback, and that correspond to the face motor representation. In macaque, we did a meta-analysis of electrophysiological and anatomical studies. And so here is the dorsal bank, is the cingulate sulcus with the fundus with the dotted line. And here in the color-coded area, you can see where we find studies finding reward or face reward-related activity or feedback-related activity in the reddish part. So all recording you can see were done in the dorsal bank of the cingulate sulcus, and you can see that this region also falls close to the journey of the corpus callosum there. Here you can see an example. And at the same time, if we look at the high face motor representation, they are almost colocalized at the same region. So in both macaque, from macaque to human, the functional organization of the mid-cingulate cortex is well preserved. So can we now better understand how the lateral frontal cortex evolved using such methodology? So I'm just giving you one example here because I have no real time. So the current view of the lateral frontal cortical sulcal organization is as follows. So in human, this is from Michael Petrides' work, you can see all these sulci that are labeled, okay? In old world monkey, we have 5 main sulci: the precentral dimple, the arcuate sulcus, the principalis. And in chimpanzee, you have 6. So you can see that this looks very different and very hard to link together. So can we go from this brain to this brain, basically, by understanding how the sulcal organization is done? So I can show you that yes, in fact, the macaque brain is more complex, and the final output is this. In fact, you find all sulci that you have in human, almost all, except this more anterior part here, but that you can find in chimpanzee. And here are all the sulci. And I'm going to show you why this principally sulcus seems in fact to be split in three and correspond to these three sulci in chimpanzee and human. So like this, these three sulci. So first, what we observed is that in macaque and in baboon, in fact, there is changes in the principally sulcus. You can see anatomical differences. For example, here in this macaque, you see there is between. So this corresponds to this difference here. You can see a little change in the sulcus here, and same thing for this between these two sulci. Here you can see there is some different shape within the principally sulcus, and it's true in baboon. And you can see here in chimpanzee, again, these three sulci, and in human. But what is very interesting is that if you look at the frequency of occurrence of these three, they are almost all present, always all present. There is no different statistical difference. And you can see that the intersection between PMFS, so the most posterior part of the principally sulcus, PMFS-E and PMFS-A, in fact, it always falls at the same level. So PMFS-E, the caudal limit is at the level of the genu of the corpus callosum, the posterior part. Here, PMFS-A, it's at the level of the rostral genu of the corpus callosum. In all species, you can see the distribution. And EMFS-H, you can see that it falls really nicely also with what we talked earlier, that is the fork with the cingulate at the end of the rostral hand of the cingulate sulcus. And we have also functional evidence of parcellation in three parts of the principally sulcus in macaque. So based on a resting state data that we got on 3 awake macaque, so we mask the principally sulcus, and based on the correlation between the Ball signal in each voxel of this mask and all voxel of the rest of the brain, we identified what was the best number of parcel, and in fact the best number of parcel was 3. As you can see here, with 4, 5, 6, 7, it didn't work well. And it gives this type of parcellation. So it's one example, but it's true for the 3 macaques and bilaterally. So you can see an anteroposterior clusterization. And if we look at where falls the point where 2 parcels are limited, we can see that the posterior one falls at the level of the rostral limit of the genu of the corpus callosum, where we find where we have this difference in the anatomy in the principally sulcus. And the most anterior one, shift here, is linked to the junction between the cingulate sulcus, the supra, and the fork. So has the anatomical landmark we find. So this is really strong results showing that the principally sulcus is in fact divided in 3 parts. And these 3 parts corresponds to the human homolog one. And also we have empirical data showing that if we look at what this PMFS-A, so the middle part of the principally sulcus, what is its function? In human, we know that it's recruited to monitor information in working memory. So this is here, PMFS-A is there. And we know in macaque that this type of function is also located in the human homologue of the PMFS-A, this second part of the sulcus. So altogether, this data strongly suggests that these regions are homologous. So to conclude, what the analysis of sulcal pattern tells us, it tells us first whether and how some frontal brain regions have evolved. And it tells us that the precise anatomo-functional organization of a given region it gives us that. And this is true in both macaque and human for the dorsomedial frontal cortex and the lateral frontal cortex. But I didn't talk about, but it's also true for the ventromedial prefrontal cortex. And so because sulci relate to function in both human and macaque, as I showed you, the anatomo-functional organization of the entire frontal cortex may be understood across species thanks to sulci. So I would like to thank all my collaborators and my lab in Lyon, and I thank you for your attention.